Okay, so good morning, everybody. Bom dia a todos. It's a great pleasure to have Dr. Stefanie Colombo from Dalhousie University, Canada, here. Thank you, Stefanie, for accept our invitation. It's a great pleasure to be with you again. And before we start, I think we can just talk a little bit for some minutes to wait the people to join us. So if you could talk us about your professional life, should be great.
So bom dia, everyone. Good morning. Thank you for inviting me this morning. It's we're in some strange times. Just a few months ago, I was with you face to face at USCI and touring the labs, and everyone was so welcoming, and I had such an amazing time. I learned so much. And a few months later, here we are doing an online webinar. So, like you, our labs are shut down, and we've had a few months of quarantine. We are just getting started again in the lab. I had about 3,000 Atlantic salmon that have been waiting for us and my grad students to get started in an experiment. So it has definitely been a challenge. We are just starting a new experiment that's a continuation of what I'm going to talk about today. So I'll talk a little bit more about that. And I have a couple of slides in my presentation about what my lab does and sort of the research direction. But I'm located in a town called Truro, Nova Scotia. It's the east coast of Canada. It's supposed to be summer now, but it's very cold. Too cold.
How about the COVID situation? How about that? How you are dealing with that in Canada right now?
I think it depends province to province. In Nova Scotia and Atlantic Canada, we've had very few cases the past month or so. So I think we're in a good situation. Other provinces like Quebec, they have a lot more cases. So if you visit another province, you go from Nova Scotia and go to another province, when you come back, you need to self-isolate for 2 weeks.
Okay.
So, and they will call you. I've just learned that they will call you every day to make sure that you are not violating that law.
That's amazing.
But they are starting to open back up again. You can go to a restaurant. Some provinces have a mandatory mask rule, so you need to wear a mask when you go inside anywhere. We don't have that in Nova Scotia, but that's a new rule around Toronto because there's many people there. So it kind of depends on where you live, because Canada is so big that every province has a different rule about how to deal with COVID right now.
That's true. It's the same in Brazil. I think not province, but any states have their own rules to try to fight back against the COVID. And the university is closed, so.
And well, we just opened back up again for 25% of the population can go back, but not for undergrad students. All of the classes starting in September will be online. And grad students, a lot of grad students are in the middle of their project. So some of them, we have to have a very particular schedule. Everyone has to wear a mask. So there are some enforcement in place to make sure that their safety is protected over the next couple of months to finish up experiments and things like that. Okay. So we've been shut down since around March 13th. And everyone's been working from home, but now we can start to slowly go back just for research purposes.
Okay. Interesting to see the differences between the countries.
Yeah. And of course, we have animals on our campus. Not only do we have all of the salmon that I'm working on, but we have cows and poultry and all kinds of animals. So people have had to go in, every day to care for the animals. So it hasn't been completely shut down because we need to make sure for the health and safety of the animals too.
Okay. Okay, so I think we can start right now. So again, welcome to our webinar. It's a huge pleasure to have you here. And well, I will turn off my camera and you can share your presentation.
Okay, perfect.
Okay.
So thanks for joining everyone this morning. I am going to share my screen now so you can see my presentation. So bear with me for one moment.
That's it.
Lila,
Yes.
you can see the presentation? Okay, great.
Yes.
So I will continue. So today I'm going to talk about some research that I've been working on for the past few years and continuing to work on it. And that is how global warming can impact the nutritional quality of seafood. So just a little bit more about my lab and what we do. Our main goal is to understand nutrition in farmed fish and to improve the sustainability and productivity of aquaculture. So we do that by looking at more environmentally sustainable feeds for the fish, such as new ingredients that can replace fish meal and fish oil. We look at fatty acid biosynthesis in fish. We use nutrigenomics as a tool to understand the metabolism of the fish, and looking at the impact of climate change on fish nutrition and metabolism, which is what I'm going to talk about today, and farmed seafood nutritional quality, because we can test all different feeds and improve the sustainability, but at the end of the day, we want to make sure that we're producing fish that are good for us to eat. So that's a little bit about what we do in my lab. So as you know, there are so many nutritional benefits associated with eating seafood. Fish is among the healthiest foods on the planet. It's high in quality digestible protein. There are so many different options for different tastes, eating food locally. There's high fat options, low fat options. Fish is also rich in micronutrients, very high in vitamin D and other micronutrients, calcium, phosphorus, other minerals. But what I'm really going to focus on today is the omega-3 fatty acids. And fish are a unique and excellent source of these omega-3 fatty acids. So just a bit more about these omega-3s. There are two main long-chain omega-3 fatty acids. There's eicosapentaenoic acid, EPA for short, and docosahexaenoic acid, DHA. And I'll mostly focus on DHA in this presentation. So for EPA, it's involved in the inflammatory response. It's a precursor to hormone-like compounds, such as the eicosanoids, and in general, it's involved in our cardiovascular health. DHA, on the other hand, is involved in our neurological health, so our brain and eye health. It's extremely important in neurological development and neurological function. Both EPA and DHA have fundamental roles in cell signaling, cell membrane fluidity, and overall growth and development. And all of these pertain to all vertebrates. So fish need them, humans need them. So they're extremely important nutrients. DHA is actually the most abundant fatty acid in our brains. So our brain is about 60% fat, and of that 60%, 10% of it is DHA. So it's being stored in there for a reason. And DHA is critical for your brain as it's developing. So there are numerous agencies that recommend DHA intake for infants and for pregnant mothers, because many studies have shown the relationship between DHA and the outcomes of the child in terms of their cognitive, physiological, and behavioral outcomes. So DHA is important for adults as well, but early on in life is when you're really setting the foundation for neurological health later on in life. So what does DHA have to do with the ocean and aquaculture? So it's actually interesting that DHA has been shown to be part of our evolutionary history in terms of the development of our brains. So through seafood consumption, DHA was involved with our brain development and has been shown to increase our cognitive ability during early human evolution. And I've provided a couple of references there. There's quite a few studies that have shown this, and I find this area of research really interesting. So you can check out those references that I've supplied at the end there. So what's unique about DHA is that it originates in the ocean. It's unique to aquatic ecosystems. DHA is not produced by plants on land. So our way of acquiring it is through eating fish and seafood. So you can see with this food chain here, DHA is produced by primary producers in the aquatic ecosystem, microalgae, macroalgae, seaweeds, and is consumed by primary consumers, secondary consumers, et cetera, et cetera. So it's specifically retained from one trophic level to the next because it's so important to the health of these organisms, including ourselves. So that's why it's retained from, the bottom of the food chain all the way to the top, is because of its importance to health. So that's an important point to note. So one of the things that DHA does is, in ectotherms, like algae, bottom of the food chain, DHA works like antifreeze, basically. Because of the structure of DHA, it's very compact and pretty bendy. It can help prevent the cell from freezing up when it's really cold. So in colder temperatures, algae produce more DHA to help prevent the cell from freezing up, the cell membrane. In warmer temperatures, algae produce less DHA because it's not needed to combat cell freezing, basically. So this idea is called homeoviscous adaptation. And many different single studies have shown, when you grow algae in cold temperatures, more DHA is produced. When you grow the same algae at a warmer temperature, less DHA is produced. So what does that mean in the context of climate change? So how will global warming affect DHA production in algae, knowing that algae use DHA as an adaptation with temperature? So higher temperatures could mean less DHA. So we knew this on a smaller scale in terms of single experiments that cultured algae, but we really didn't know this on a larger scale. So a couple of years ago, we conducted a study that looked at the temperature influences on DHA production in algae. So we did a data synthesis. We collected over 1,000 data points on algae from the literature, and we did a simple regression. We just looked at how does increasing temperature impact the proportion of DHA produced by algae, and we looked at different species of algae in different parts of the world. So we were trying to go larger scale with this. And what we found was after 16 degrees, algae do actually produce less DHA. And what we estimated with this model is that 28% less DHA would be produced with a 2.5 degrees Celsius increase in water temperature. And I provided the reference for this study as well. So knowing this in algae and knowing that DHA is selectively retained as you go up the food chain, what does that mean for the fish that we're eating, that we're getting our DHA from? So that was the next question we asked, was how will this predicted decline in DHA affect fish? So if we know that DHA is transferred and accumulated and there's 28% less DHA at this point, how will that increase, or how will that decrease with higher trophic levels? So really the question is, what's going to happen to our supply of DHA that we get from seafood? So our question is, how much DHA will be available in fish? So first question is, how much fish are we even harvesting every year? So we had to answer that question. So we looked into the literature at marine and freshwater fisheries and aquaculture about what's the total catch every year. Of that catch and the edible portion of the catch, how much DHA is harvested from those fish? And so the next step is, based on the model from algae, what would be the estimated decrease in DHA in fish from now until 2100? And the year 2100 is based on the predictions from the Intergovernmental Panel on Climate Change. And their model, the RCP 8.5, which used to be considered an aggressive model for greenhouse gas emissions, but now it's just considered the business as usual scenario, which is an average 2.5 degree increase in water temperature. So what we're creating here is a new model for fish based on what we know in algae. So remember, first we had to know, how much fish are we consuming every year? How much fish are we harvesting? We got that data from the Food and Agriculture Organization of the United Nations, and also the Fishery Research Institute called the Sea Around Us, to get numbers on how much edible fish is caught and farmed worldwide every year. Then we had to determine how much of that fish is contained DHA. And we got that from some previous models that myself and others have published. So based on those numbers, we came up with a base case of how much DHA is produced every year for human consumption. Then what we had to do was look at the temperature influence on DHA production on algae. So we used that model from the previous algae data synthesis. So then we determined roughly how much DHA is presently available, and then predicted how much DHA will be available in 2100, so 80 years from now, based on the current models of climate change. So this is actually showing the amount of DHA available from fish for human consumption right now. And the different areas and boxes represent the United Nations fishing zones. Some of them are by country, and some of them are divided by the fishing zone that's allocated by the UN. So you can see there's a difference right now in how much DHA is being harvested. This is based on the species of fish. It's based on the temperature of the water where it's harvested. So there's already kind of a disparity on the amount of DHA that's produced in each fishing zone as it is. So that was interesting. So what we learned right now is that we harvest about 370,000 tons of DHA every year. And this is in line with other predictions that have estimated how much DHA is produced every year. So we knew that we were in the right ballpark of how much DHA we produce right now, or that we're harvesting right now, rather. So based on the algae model, we applied that to fish in different fish zones based on temperature. And on average, we found that the 2.5 degree increase in temperature could result in a 58% reduction in DHA globally. That number depends on the geographic region and how temperature will be impacted and what's the amount of DHA produced in that zone. But on average, it was 58% decrease by the year 2100. So that means it's predicted to decrease to about 140,000 tons of DHA from 370,000 by 2100. So that, for us, myself and co-authors, to learn that was a pretty jarring number. So this is again showing by fishing zone that this is the percent reduction in DHA by 2100. So this is not amount of DHA anymore. This is the percent that we predict it will decrease. And so the darker red colors are very high decreases, over 80% reduction in DHA. And the lighter colors mean that there's less reduction in DHA. And there's actually no data for Antarctica in terms of fish catch and temperature. So we could not do the South Pole regions. So, you know, in North America and temperate regions are actually worse off in the decline of DHA, and I'll explain why in a minute. South America is a little bit better off, and Asia is sort of in the middle. So one of the reasons why some of the temperate regions is because of the freshwater fish in temperate climates, and we predicted those to have the greatest proportional decline in DHA. And one of the reasons for this is that there's a larger predicted temperature increase in freshwater than in oceans, because they're smaller bodies of water, so they're more susceptible to rising air temperatures. Asia has the greatest catch and supply of freshwater fish. It also has the greatest share of the world's population. And I'll show you in the next slide. We actually looked at the predicted DHA based on per capita. So in thinking about per capita, larger countries with a very high population or estimated population growth will face more severe DHA shortages because there's more people, especially in those regions that are depending on freshwater fish as their main source of DHA. So right now, today, the global per capita, based on the UN's estimate of the global population, is 140 milligrams per day of DHA. Everyone gets 140 milligrams per day. So for infants, the recommended threshold is 100 milligrams per day of DHA. So we're good for at least supplying infants with the minimum requirement. But the other recommendations for adults are actually 250 to 500 milligrams a day. So right now we're probably not even getting enough or producing enough DHA to meet that requirement. But so given the importance that we know DHA is to infant brain development, we kind of focused our study on that. So given the actual distribution of DHA production, we estimated that right now 58% of the world's population currently live in countries where DHA availability from domestic fish is already below this threshold. So if you consider that half of the world is not getting enough DHA already, it's not really the best situation in 80 years from now when we expect DHA to decrease due to global warming. So this is the current estimate of DHA production in milligrams per day by country. This is no longer fishing zone. This is by country. And so you can see the really dark blue is over 250 milligrams per day. So that's actually meeting the adult requirement. And you can see some countries like Chile, Greenland, Norway, will be reaching the current estimate of DHA production, whereas North America and US is already not reaching that threshold, and Brazil is good. This is in 2100 applying our model, and anything below 25 milligrams per day is in red. That's a lot of the world. Some countries are still not really affected, and there's a reason for that. So the countries that are in dark blue are not affected by that as much. And the reason for that is that they have large fish production already and very low or relatively low populations like Greenland, Norway, Chile, New Zealand. They're all above 250 milligrams per day per capita in both the current model and the predictive model. And some countries still remain above the 100 milligram per day threshold. North America and most of Western Europe are still below this threshold now and in year 2100. But that wasn't as much concerning as it is for certain populations. So you can see there's a disparity in which countries will have the most available DHA versus others. And what was, I guess, more concerning to read is that the available of DHA in 2100 is lowest globally in some African countries, actually most African countries. So DHA production in inland Africa could decrease by 65%. And there's only one exception, and that's Namibia. And so most African countries will produce less than 25 milligrams per day of DHA per capita. So the reason for this is that the UN predicts a very large population increase for African countries by 2100, and the temperature-driven declines based in these areas. So this is concerning for developing brains, the fetus and infants. And this is a concern in developing nations and tropical regions where human nutrition depends most on wild fish. So the recommendations we know for DHA can vary. So it's 100 milligrams per day for infants, but we also know that for adults, it's over 250 milligrams per day. And our study is not the only one that's concerned with the impact of climate change on nutrient production in seafood. So for example, a study that came out a couple of years ago by Chris Golden and colleagues estimated that 19% of the world's population is vulnerable to nutrient deficiencies, and that they included DHA in that study due to declines in fish catch caused by overfishing and climate change. Hicks et al. Last year found that several countries where there's micronutrient intakes are inadequate. The nutrients available from fin fish catch exceed the dietary requirement. So again, it's just showing the importance of fish consumption to especially growing children. A couple of years ago, Cheng et al. Found that there's a predicted decrease in fisheries catch, global fisheries catch up to 13% by 2050 in a high emissions scenario, which, like I said, is the business as usual scenario now. And finally, Myers et al. Stated that global food production is likely to be altered through several climate change related pathways, which ultimately can affect the quality of our food that's produced in agriculture, fishery, and livestock, and aquaculture. So there's a lot of research coming out recently that's showing that climate change can affect our nutrient production. So what does this mean? Does this mean that we can expect a decline in our brain health for future generations? That's a scary thought, but it's not all doom and gloom. There's a way to bridge the gap, and there's hope. So aquaculture has a huge role to play in producing our seafood. Currently, right now, we know that aquaculture supplies about 50% of our seafood right now, and this is estimated to decrease. So what the trick of this is making sure that DHA is supplied in the diet of the fish so that the DHA is stored in the fillet and we're getting an adequate supply. But the problem right now is that we need new sources of DHA, because currently the main supply of DHA in fish feed is wild sourced fish oil. And this is a huge problem. So if we're not using fish oil in the diet, we're declining the DHA that's being stored in the fillets. But the good news is that there are a lot of new sources that are being developed, both for human consumption and for aquaculture feeds. Some examples of this are microalgae and other microbial organisms, genetically engineered oil seeds like canola, yeast, other single-celled organisms. So there's a lot of new sources that some of them are becoming commercially used, not only just to supply DHA, but other sources of micronutrients if we are not to use wild sourced fish oil in aquaculture feeds anymore. So this is a positive thing. So just to sum up, DHA is very important for our brain functioning and development. And it is concerning to learn that by 2100, we could have 50% less DHA that's naturally produced by algae, and then that's stored in fish, so we could have less access to it. And this could be potentially a problem in vulnerable populations that depend on the fisheries as a main source of DHA, especially for infants. But I think the good news of this story is that we have an opportunity to produce fish and a healthy source of DHA through aquaculture, to supply an adequate source for our future generations and their brain health. So this is the actual paper that we published in AmBio, the journal AmBio, last year. So I put a link there. You can check out the article and all of the nuances of the methods of the model. And there was some press associated with the article, and I really liked this article that was written about the study in the section of neuroscience in Scientific American, and I provided the link there so you can check it out. So thank you to my lab, to the grad students in my lab. They are, like you guys, are affected by the pandemic. And my message to them and to you is to, don't give up and keep going with your studies, despite, the challenges that we face right now in the world. And to the co-authors of my study, the Faculty of Agriculture and the Natural Science and Engineering Research Council for the funding for this study. And so the study that I just talked about, we're continuing, and we are testing the model in the lab because, that was based on a data synthesis. So we do actually want to know that if we increase the water temperature and we decrease the DHA in the feed for the fish, what will actually happen? Are the fish going to synthesize more if it's not in their diet? Are they going to need less because the water temperature is warmer? We're not sure. So we actually just started that experiment this week. So hopefully I'll have some more results later. So those are the references that you can check out if you're interested. And I'm going to stop sharing my screen.
Thank you, Stefanie. It was a wonderful webinar. Now we can say webinar.
Yes.
And thank you for sharing that. This is amazing because we have a group of Débora Fracalossi that is working as well with DHA with tilapia in the south of Brazil. And there is a lot of students of her that I think they are here. So we can open now for the questions. And I'm sorry, Stefanie, I will talk in português.
Go for it.
E se vocês, né, não quiserem falar em inglês, podem abrir os microfones e fazerem a pergunta que eu faço a tradução, tá? Podem ou levantar a mão ou se inscreverem, abrirem os microfones, só não pode falar todo mundo de uma vez, tá? So, I don't know, anyone? I will check in the YouTube as well. Oh, Delano,
I'll just say I'm sorry if you could hear like a lawnmower in the background. And in my presentation, I went and shut the window because I thought maybe it was so noisy. It's so hard working from home sometimes, you know, because unanticipated noise.
I know. Every time that I am talking with somebody or I am in the meeting, somebody called me because there is a delivery or something. But at that time, they always call me. And this is terrible.
Yeah.
So Delano is watching us in the YouTube. Delano said, "Great talk, Stefanie."
Oh, Delano, thank you.
So I don't know. Anybody? No, nobody. I have a question, Stefanie. Maybe. Just to tell to everybody that we have a joint project together, investigating the seaweed as ingredients, and of course we can increase that. But I think one of the option, of course, is improve. I'm trying to not be tenacious here, but maybe is a good argument to now people think about the cultivation of algae in general. Of course, microalgae is much more have much more DHA than seaweeds, and seaweeds have another potential for the part of the immunostimulants and something like that. But I think more and more is important and urged to start to thinking about the aquaculture and put more attention in aquaculture of algae in general, because this data that you present is very scary. So
yeah, algae is going to save the world.
Yes.
Macro and microalgae will save the world. But I mean, it's true. Like macroalgae is not going to be a huge source of DHA, but it does supply DHA, and not only just for, you know, using in fish feed, but for direct human consumption.
Yes.
But also there's sort of another roundabout way of thinking of it, is that algae can help improve the gut health of salmon and other fish. And so if the gut health is improved, it means that their uptake of other nutrients like DHA in the diet can be improved, and you're not wasting anything.
Yeah.
So, I mean, that's just thinking of it in terms of DHA, but in terms of using macroalgae in feeds for finfish, prebiotics, especially in relation to health, which is, I didn't even talk about that. That's a whole other issue, is fish health. So macroalgae has a very important place, an important role to play in the future of aquaculture feeds, for sure. Yeah. And my work tends to focus on salmon and rainbow trout, but there are so many other opportunities. Tilapia would be a perfect example.
We need to have a start point. So I think it's okay. It's fundamental. Then Canada start with salmon and trout, rainbow trout. Yeah. And for our side, I think is the tilapia, because it's the first freshwater fish that is commercialized now in Brazil, and the market is growing on. So, oh my God, everybody's shy. Somebody? No? Nothing? You can open the mic. Oh, I have a question to do. Okay, Simone, go on. You can open your mic if you want.
Hello?
Hello, okay.
Can you hear me?
Yeah, perfect.
Yeah.
Oh, okay, good. Hi, Stefanie.
Hello, Simone.
Congratulations for your presentation. It's very good. I like too much.
Thank you.
Yeah, it's a good theme. It's important point of view to do prospection of the DHA production. And we are also working with shellfish, and we are looking in a doctor degree thesis. I don't know if Luciani is here, is watching us.
I don't know.
Yeah, in her study, she will look for DHA production in mussels, and yeah, we would explore this theme. And I would like to ask you if you are using mussels or shellfish in your study.
That's a great question. And we didn't include shellfish at all, which does represent another source of DHA, which was not included in our data synthesis. So, I mean, shellfish do supply DHA. We didn't include because that would make the data set even more humongous than it already was. But the DHA that's supplied, like quantitatively, is lower than is what's supplied in fish. So it probably won't change the situation or our prediction too much. But it is a very good point, also because that what shellfish supplies is an extremely sustainable source of DHA. So it shouldn't be ignored. We do have a paragraph in our study that's just dedicated to talking about the DHA supplied from shellfish. So it's a really good point. But I would be interested to see if our model also applies to shellfish as well. So if the algae is producing less DHA, will that impact them, or will they synthesize more DHA if they don't have access to it? Those types of questions, sort of like what we're testing in the lab right now in fish.
Yeah, this is a very interesting, and perhaps, I don't know, I just was thinking we can share results, and because we are monitoring mussels in the environment from farmers, and then I don't know, it's just an idea.
Yeah, that sounds really interesting. Leila, maybe you can connect us.
Yes, for sure. For sure. Simone is our invited professor in the program, so and she spent some time in Canada as well. So
really?
Yes.
Yeah, I was in 2011 in Nanaimo at VIU.
Oh, really? Okay.
Yeah, I was working with Helen Gurner-Smith. I don't know if you know her.
Did we meet each other?
I don't know, perhaps.
Okay, I'm just wondering. Yeah, anyways. Okay, yeah, I know Helen. I know Helen, and I have been to VIU, so that is a really nice place to work for a little bit.
That's funny.
Yeah, she's now near from Toronto, I think, and also she's mom of two now.
Yeah, well, she's actually in New Brunswick at Huntsman. But so yeah, she went east. She went east for sure, and she's now in New Brunswick.
Okay, Simone, we are cool?
I'm here, I'm just listening.
No, okay, it's okay. So, do you have any questions?
No, I'm okay now.
Okay. Stefanie, Delano have a question here in the YouTube. So, for animal feed, including higher levels phospholate in the diet, could increase the DHA absorption and deposition? This is one, and there is another one. But you can answer that and I can come back.
Okay. I mean, that is definitely a big concern with using plant-based diets for fish, is because the plant oils contain very little, if not no, phospholipid. And so that is a concern in terms of phospholipid requirements. But it's also a concern for, like you said, the deposition of DHA. Another thing that I've been learning about more recently is, because there's no phospholipid, there's less choline, phosphatidylcholine. And even though choline is not a required micronutrient, it's showing relationships with fat deposition in the intestines, and this is not good. So if there's no choline, which is usually supplied by phosphatidylcholine, then there can become a problem. So that's why we need to think of sources that have phospholipid, and even though it could supply a plant source of DHA, it's not containing phospholipid, so this could be a problem.
Okay. And the second question is, in the case of the future seaweed production in Santa Catarina, Brazil, Delano, do you think it will be better to harvest seaweed during the winter, aiming to meet the DHA demand?
Yeah, I get it. I get what he's saying. That's a really interesting question, because I assume that, because our whole study, both of them, the algae one and the fish one, was based on microalgae. I would assume that, they have the same relationship with temperature, that in colder temperatures they produce more DHA, and warmer temperatures they produce less. But I'm not sure, because we didn't do that first analysis with macroalgae. So I can't really say, but if that's true, then probably the better time would be during winter, when the temperature is colder. There should be some more DHA. Mm-hmm. But because we know macroalgae doesn't make a whole lot of DHA, I'm not really sure how much that difference would be. But it's an interesting question. That could be another data synthesis paper of just collecting fatty acid profiles for macroalgae at different temperatures to see what's the relationship.
Yeah, this can be interesting. But seaweeds don't have DHA, until I know. But
some do, but very low. Some do, but very low. Yeah. So I'm not sure how much that would make a difference, really.
Yes. I think Kappa Ficus doesn't have, so my baby doesn't have. Okay, I think that's it. Nobody else here in the program? Felipe, go ahead.
Can you see me now? Yes, you can see.
Yeah.
Yep.
I know why Delano asked that, because we recently have an experiment, and we included uva in the diet of marine fish, the jurel. Don't know if you know the jurel. It's kind of a tuna from Mexico.
Oh.
And the DHA level in the fillet of the fish increased with the inclusion of the uva on the diet.
Huh.
And the uva does not have DHA, a lot of DHA. So we are kind of trying to understand why that happened, because it was very interesting, and it was significant, and it was around a 35% increase in the DHA level in the fillet.
So, I mean, was the tuna like synthesizing? Does it have high alpha-linolenic acid? Is it high in omega-3, 18 omega-3?
No, we included just 1% of uva in the diet, was just a little.
Weird.
I can send you the data after. I will send you.
Yeah, I'm really curious.
We need some help indeed to try to interpret that, because my clue is bacteria, associated bacteria, but I really don't know. What can be
bacteria, or what was the other lipid sources? Where were the lipid sources in the diet?
It was fish oil. inefficient.
Fish oil, like somehow improved bioavailability, but at 1%, how?
Yes.
That's super interesting.
The idea was just evaluate the action of the uva, the seaweed, as ingredient, bioactive compound, not as ingredient, but as bioactive compound. So we included just a small portion.
Wow.
And
that's amazing. A nice little feed additive.
So our next project just put in the paper that we can test that as well.
Did you, I mean, I probably wouldn't have thought to look at this either, but did you have any samples of the gut microbiota you could check out?
I'm not sure about that. We were asking Esmeralda. Esmeralda is the one that is this project.
Super interesting.
Yeah. Okay, so I think we are good. So nobody have any other questions? Just to remind everybody that Stefanie is our partner here. We have a joint project together, and I hope we can. This project will finish. It would be finished this year, but after the pandemic, I think we will have a extension until next year. And we are preparing the second project to some call that we have. So Stefanie, students that want to go and see what Stefanie is doing in Canada, just talk to me, and we can try to, after the pandemic, of course, we can try to make this happen. Okay, Stefanie, it's a great pleasure to have you here. I always say this to you, but it's a great pleasure to have you as a partner and now as a friend. And I hope you have a lot of success in your work. And if we can help you on this, it will be great. And just take care of yourself here. And thank you again for coming.
Thank you. Thanks for inviting me. Thank you, everyone, for coming. So hopefully I'll see you in Brazil next year.
Hope so, fingers crossed. Thank you.
Okay, good. Stefanie. Have a great day, everyone.
You too.
Bye-bye.
Thank you, everybody. Bye-bye.